Good morning children, I am Sargaisis. Now I am going to teach you mutation. Mutation is the sudden heritable change in the genotype of an organism. This mutation causes alteration in the DNA. So along with the genotype, phenotype also get changed. So here the mutation is the sudden heritable change in the genotype of an organism. So that leads to the change in the phenotype also. So the individual which are showing a different phenotype or change in phenotype due to mutation are called mutant. So mutant are individual which are showing a change in the phenotype due to mutation is called mutant. And the factors which cause mutation are called mutagens. So mutagens are the factors which cause mutation. Is it clear about mutation? Let's see which are the factors that cause mutation or which are the factors that act as mutagens. So there are two types of mutagens, physical and chemical mutagens. So the physical mutagens are X-rays, UV rays, etc. So such radiations cause a change in the genetic makeup of an organism, and that is the physical mutagens. And chemical mutagens are some chemicals like nitrous acid, mustard gas, then EMS, ethyl, methane, sulfonate, etc. are the chemical factors. Is it clear about the mutagens? Next we can see which are the types of mutation. Okay, the types of mutation. Mutation is of two types. They are gene mutation and chromosomal mutation. These are the two types of mutation. Here, this gene mutation again classified into point mutation and frame shift mutation. Okay, and this chromosomal mutation again classified into chromosomal aberration and ploidy. Okay, so chromosomal aberration is the variation in the structure of chromosome. Ploidy is the variation in the number of chromosome. Okay, and this ploidy again classified into two. Aneuploidy and euploidy. Okay, these are the different types of mutation. Is it clear about the types of mutation? Now, next we can see one by one in detail. First, we can see gene mutation in detail. So here, gene mutation. Gene mutation means a sudden heritable change in the single base pair of the segment of DNA. In the segment of DNA, there is a change in the single base pair, and that mutation is called gene mutation, or single gene get changed. Okay, and this gene mutation is of two types, that is point mutation and frame shift mutation. Here in this point mutation occur due to the change in the single base pair. A certain point of the base pair get changed that cause mutation. Okay, and example, sickle cell anemia. cell anemia is an example for point mutation. So let's see. So normally the RBC is spherical shaped. So due to the change in the single base pair, actually the glutamine is replaced by valine in the DNA segment that I will teach you in the next chapter. So here the spherical shaped normal RBC change into sickle shaped. So this condition is called sickle cell anemia, and after this leads to anemia. Okay, so that is sickle cell anemia. Here next frame shift mutation. Frame shift mutation means the mutation occur in the base pair due to the deletion or addition of base pair. If any base pair is added to the segment of DNA, or if any base pair is removed or deleted from the segment of DNA, that cause frame shift mutation. So that cause change in the reading frame of the segment. Okay, so that is called a frame shift mutation. Example, thalassemia. Thalassemia is an example. This is a condition, or this is a type of disease, a disorder due to this frame shift mutation. The disorder is the body cannot make normal hemoglobin in this thalassemia condition. So that results in the excessive destruction of red blood cells, RBC. Okay, so which leads to anemia. Is it clear? So this is the condition of thalassemia, and that's all about gene mutation. And next we can see the chromosome mutation. Chromosome mutation. So in chromosome mutation, the mutation occurred due to the change in structure and number of chromosomes. Let's see, if there is a change in the structure of chromosome, that mutation is called chromosomal aberration. And if there is a change in the number of chromosome, that is called ploidy. So here, this is structural variation. And here this is numerical variation or number variation. Numerical variation, that is the ploidy. So numerical variation of chromosome is called a ploidy, and the structural variation is called a chromosomal aberration. This is the reason for chromosomal mutation. First we can see the chromosomal aberration. As I told you, the chromosomal aberration is the change in the structure of chromosome that leads to mutation, and that mutation is the chromosomal mutation. Example, we can see this chromosomal aberration in cancer cells. Okay, so the cancer cells are showing this chromosomal aberrations. And next is the ploidy. Ploidy is the change in the number of chromosomes. As I told you, the ploidy is of two types. They are euploidy and aneuploidy. Euploidy and aneuploidy. Euploidy is also known as polyploidy. Okay, so we can see the euploidy and aneuploidy in detail. Okay. Next we can see the euploidy. Euploidy is also known as polyploidy. Here, the mutation occurred due to the change in number, and that is exact multiple of the basic number of chromosomes. Example, so normal it is 2n. So if it is multiple means 3n or there is a change in the number of chromosomes, and that change means exact multiple of the basic number of chromosomes. That is polyploidy or euploidy. But in case of aneuploidy, here it is not an exact multiplication, it is the addition or deletion of one or two chromosomes. Okay, next we can say this aneuploidy is, for example, 2n plus 1. That means along with the 46 chromosomes, we can see one extra chromosome. So total 47 chromosome, and that condition is called trisomy. Okay, so normally 2n means what? 46. That is 23 pairs. So 2n is the normal basic chromosome number, and along with the normal basic chromosome number, there is one extra chromosome, so total 47, and the condition is called trisomy. This is the change in the number of chromosome. And another condition is there, if 2n minus 1, that means what is the actual number of chromosome? 45 only. So that condition is called monosomy. Monosomy. And another condition is there, that is 2n minus 2. That means one pair chromosome is missing there. That means only, what is the number? 44. So that is called nullisomy. Nullisomy. Here one pair is deleted, one pair is lost from here. That is only 44 chromosomes are here. So this is called aneuploidy. So aneuploidy is the three types: trisomy, monosomy, and nullisomy. Is it clear about the chromosomal mutation? Next we can see pedigree analysis. The ancestral history of an individual is called pedigree. Pedigree analysis means the analysis of a particular trait in several generations of a family. Here, the particular trait can be analyzed in several generations of the family. Then the expression of that particular trait is assembled in a family tree, and that is called a pedigree analysis. Here are some symbols used in pedigree analysis. Let's see the symbols first. So pedigree analysis, as I told you, pedigree is the ancestral history of individual. Pedigree analysis is the analysis of a particular trait in several generations of the family. Okay, and now we can see the symbols. These are the symbols used in pedigree analysis. So this symbol represents male. Okay, square represents male, and this represents, this particular diagram, this circle represents female. The circle represents female. Okay, and this one diamond shape represents sex unspecified. Sex unspecified. Okay, and here this is a male, this is a female, and this is the unspecified sex. These are the infected or affected individuals. Affected individuals. For example, if a male is having dark shades, means this is the affected male. Okay. And here, this male marries a female, or we can say mating between two individuals. Mating between male and a female. That is called or marriage between two individuals, male and a female individuals. And this is the mating between close relatives. Mating of the relatives, close relatives. Next, we can see parents and next generation. So, the parents are here, they are having children. Okay, parents above and children below. Parents and offspring. Offsprings. Here, the male parent, this is the female parent, and they are having one daughter and one son. Next symbol we can see here. This parents, male parent and female parent, they are having only one son, and that son is affected. Okay, so parents with the affected son. That is the meaning of that symbol. And this is the children, the 5 children with the or unaffected 5 offspring. Unaffected 5 offspring. Okay. So these are the symbols used in pedigree. Is it clear about the symbols? Okay. Now we can see, as I told you about the analysis of a particular trait, whether that trait is expressing in any generations of this family, that particular family, that can be analyzed through this pedigree analysis. And normally the traits are of 2 types. Autosomal traits. Trait means characters. Autosomal traits and the sex-linked traits. And the autosomal traits again classified into autosomal dominant trait and autosomal recessive trait. Let's see an example of an autosomal dominant trait in a pedigree chart. Okay. So here, before that I want to tell you about the traits. The traits are of 2 types, autosomal traits and sex-linked traits. So the autosomal traits are also classified into autosomal dominant trait and autosomal recessive trait. So here we can see these 2 types of autosomal dominant and recessive type examples as a pedigree. Here the autosomal dominant trait example is myotonic dystrophy. Myotonic dystrophy is a condition in which there is a weak skeletal muscle. That means due to the deficiency or any defect in the muscular protein that cause the death of the muscle cells or tissue. That means weak skeletal muscles. That is, the condition is called myotonic dystrophy. Here in this pedigree, you can see an affected female parent, then a normal male parent. And they are having 4 children. These are the children. 1 girl, 1 boy, 2 boys, then 1 girl. These are the 4 children. And here the daughter is free from the disease, but 1 son. So this son is affected. So he has myotonic dystrophy and he is also free from the disease, and this daughter also affected. So out of these 4 children, 2 children such as 1 son and 1 daughter are suffering from myotonic dystrophy, just like the female parent. So this is a dominant trait. So this is expressed in the parental generation. Here this unaffected son marries an unaffected lady. So they are having 3 children. They are free from myotonic dystrophy because the son is also free from the disease. Okay. Here that affected daughter marries an unaffected male, but in the offspring we can see 1 daughter is suffering from the same disease as the female parent has. Okay, so here we can conclude that if one of the offsprings is having the problem of myotonic dystrophy, that will definitely appear in the offspring because it is a dominant trait. Is it clear about this pedigree chart of myotonic dystrophy? Next example is autosomal recessive trait, example sickle cell anemia. Sickle cell anemia, I taught you, the normal RBC becomes sickle shaped and that leads to anemia. Moreover, this sickle shaped RBC cause block in the capillaries. And the details of sickle cell anemia I will teach you later. Okay, in the genetic disorder I will teach you. Here, a normal male and female got married. Then they are having 5 children. So one male unaffected, one son affected, and one daughter unaffected, but one daughter affected, and one son unaffected. So these, out of these 5 children, one son and one daughter, they are suffering from sickle cell anemia, and that means this is a recessive trait. So here the parents are having this sickle cell anemia causing gene as the recessive trait. They are not expressed in that generation. That means that gene is hiding there in one of the parents. And in the next generation it is expressed because it is a recessive trait. When 2 recessive traits bring together, definitely that will express. Okay, so here as a recessive trait in the next generation that will express it, one daughter and one son. Here also this daughter is free from the disease, but in the next generation when she got married to a normal male, but out of 3 children, one son is affected. That means this daughter must have the sickle cell anemia. That means that sickle cell anemia causing genes are hiding there. As it is a recessive gene, she is not expressing the disease because that is only a recessive gene. And this recessive gene is hiding here in this daughter. When in the offspring, when 2 recessive genes bring together, that express in the son. Is it clear? So this is the autosomal recessive gene. Hope you understood everything about mutation and pedigree analysis. That's all for today. Thank you.